Hello boys, welcome back to another session of online biology video tutorials. And this session is meant for class 11th biology students, and this is for 19th July 2021. As we have been discussing about chapter number 3, plant classification. And in this plant kingdom, we were talking about basically the fundamental basis for classification of all the plants. We said the basis is plants having seeds and plants having no seeds. So cryptogamy and phanerogamy. This system of classification is proposed by Eckler in 1883, and this is the basis for the classification of plants. And further, we said that under cryptogamy, we talked about 3 subgroups, that is algae, bryophytes, pteridophytes. And under phanerogams, we talked about gymnosperms in our previous session. And in this session, we are going to talk about angiosperms. Now, the moment you listen to the word angiosperm, you must remember that these are the plants having seeds, and the seeds are covered. So, covered seeds plants. And as we say, they are the most beautiful flowering plants. We go ahead with the morphology and the habitat. They are exceptionally large group of plants. They range in size from small, tiny, almost microscopic, like Ulfia. You can see here, they are just like small spherical structures, to tall trees such as eucalyptus over 100 meters. They are divided into 2 classes, dicotyledons and monocotyledons. As the name suggests, cotyledon means covering. If the seed has got 2 coverings, then we say dicotyledons. If the seed is covered with only 1 seed cover, we say monocotyledons. So, the seeds that have 2 cotyledons, there are 2 cotyledons in the seeds. As you are seeing here, the seed coat, then endosperm, and cotyledon 1 and cotyledon 2, 2 cotyledons. So dicot we say. And this is how the seed germinates, as we are seeing in different phases. So there are 2 parts. One is the radicle, another is the plumule. And they are having beautiful flowers, and flowers vary with different dimensions. Some of them are tetramerous. Tetra means 4. So there are 4 members in each floral whorl. As all of you know, in any flower there are 4 whorls, means round structures. The structures that are arranged in the form of a circle. So first whorl, as we talked of, was the calyx. Second whorl is the corolla. Third whorl is androecium. Fourth whorl is gynoecium. So here, when we say tetramerous, calyx and corolla, there must be 4 sepals and 4 petals. When we say pentamerous, there should be 5 petals and 5 sepals. Along with that, when we take the vertical section of the ovary, we can see the presence of locules, either 4 locules or 5 locules. So based on the number of petals, sepals and locules present inside the ovary, we say trimerous, tetramerous, pentamerous, and of course polymerous also. And they have, in monocots, on the other hand, only 1 seed covering will be there, 1 cotyledon. As you are seeing here, there is single cotyledon here. So since it is having only 1 cotyledon, we say monocots, monocotyledons. So in this, you can see on the leaves parallel venation, whereas in dicots it is reticulate venation. And then if you see the flowers, trimerous means 3 petals, 3 sepals, 3 locules like this. Okay, 3 members in each floral row. Calyx 3, corolla 3, androecium 3, gynoecium 3. So 3 number, that is the reason why we say these flowers are trimerous flowers. And reproduction, of course, since they are all flowering plants, flower is the reproductive structure. Why is it so? Because flower bears male sex organ called stamen and the female sex organ called ovary or pistil. In fact, pistil or carpel we say. So since the flower is bearing the sex organs such as stamen and pistil, we say flower is the reproductive unit or reproductive structure of the plant. And in flowers, of course, there are varieties of flowers. A flower having only stamens is called the male flower. The flower that is having only pistil or carpel, female flower. And then there is a bisexual flower. A flower having like hibiscus, both stamen as well as pistil. So there are 3 types of flowers. So male sex organ in a flower is stamen, and each stamen consists of a filament, this one filament, a tube-like structure, and anther at the tip, this lobe anther, this structure. So when the anthers, within the anthers, when we take the longitudinal section, within the anthers, we can see the pollen mother cells, and each pollen mother cell undergoes meiosis to produce microspores, and each microspore matures into pollen grains. This is how the male gametes are formed in a flower. Whereas in female sex organ is the pistil or carpel. It consists of a swollen ovary at the base, a long slender style, tube-like one, and then stigma, okay, having a lot of chemicals. And the ovary inside contains ovules, which are going to develop into future seeds. And a ovule has a megaspore mother cell. So, megaspore mother cell, MMC. And each MMC of the ovule undergoes meiosis to form 4 haploid megaspores. See, there we said microspore. Microspore is getting matured and forming pollen grain. Here megaspore is becoming matured and forming the female gamete. So out of the 4 haploid megaspores, 3 of them degenerate and 1 divides to form embryo sac. This is important. Only 1 is the functional megaspore. As you are seeing here, the carpel consists of stigma, style, and bulged portion ovary. Inside the ovary, ovule, and inside the ovule, MMCs. And MMC will undergo meiosis to form haploid megaspores. And functional megaspore will give rise to form a embryo sac. So basically, that one megasporangium, okay, divides and forms megaspore tetrad, and this megaspore tetrad will form an embryo sac. So other 3, what happens to other 3 megaspores? You can see here they get degenerated. Only 1 will be the functional megaspore, and that will have integuments which will be covering at chelagel end, and meiotic tetrad will be there. Finally, the role of functional megaspore that gives rise to embryo sac. Each embryo sac has 3 celled egg apparatus. 1 egg cell, 2 synergids. Now you can see here the presence of 1 egg cell, okay, and 2 synergids, left and right, at micropylar end. So this is called egg apparatus. So basically, in a matured embryo sac, an egg apparatus will have 3 cells. What are those 3 cells? 1 egg cell and 2 synergids. And just below that, there will be 2 polar nuclei. This is the polar nucleus 1, polar nucleus 2. And then this polar nuclei eventually fuse to produce secondary nucleus. Okay, these will form one cell. And then at the other end, that is chelazal end, there will be antipodals, 3 antipodals. Overall, if you see in a mature embryo sac, there will be how many nuclei? 3 plus 2, 5, 5 plus 3, 8. 8 nuclei. That's why we say a mature embryo sac will have 8 nuclei. But how many cells? Antipodals 3, and these polar nuclei join together and form one cell. Okay, that is considered as one cell with 2 nuclei. And this egg cell and 2 synergids. So 3 plus 1, 4. 4 plus 3, 7. So 7 cellulate. So we say an embryo sac, when it is matured, it will have 8 nucleate, 7 celled. And how this embryo sac, mature embryo sac is formed? And the functional megaspore divides, forms 2 nuclei. 2 nuclei will further undergo mitosis and form 4 nuclei. 4 nuclei further undergo mitosis and form 8 nucleate. That's how the 8 nucleus will be present. What about life cycle? Life cycle, basically angiosperm is a sporophyte, and the sporophyte will be having a flower, and the flower will be having the gynoecium part of it, having a carpel, and the carpel will have 3 parts: stigma, style, ovary, and inside this ovary, ovule will have embryo sac, and the embryo sac will have egg. So egg or ovum is the gamete, female gamete. On the other hand, the flower will have androecium. Androecium will be in the form of anther. Anther will have pollen grains, and the pollen grains are the male gametes. So there is presence of microsporangium. So microsporangium, each microsporangium will undergo meiosis and form 4 functional microspores. So what is responsible here is meiosis. Okay, and on the other hand, the megaspore mother cell present in the megasporangium of the ovule will also undergo meiosis, but instead of 4, only 1 functional megaspore will be formed. Only one functional megaspore. Now the fusion. Out of the 4 or many microspores that are formed, they just get into the pollen grains, and the pollen grain now meets the egg, and in the pollen grain, when it falls on the stigma, it generates pollen tube, and by the time pollen tube is formed inside the style, there will be 2 nuclei, that is nuclear division takes place in the pollen grain, forming pollen grain 1 and pollen grain, inside the pollen grain, male nucleus 1, let me write here, male nucleus 1 and male nucleus 2. So there are 2 male nuclei. So male nucleus 1 fuses with the nucleus of the egg that is present at the micropilar end along with the synergids. So as a result of fusion of male nucleus 1 with egg, nucleus of the egg or ovum, results in the formation of zygote. This is, we call it syngamy or fertilization 1. And the zygote now develops into embryo. Embryo will develop into an adult sporophytic plant. So this is how the life cycle of angiosperm takes place. What is the fate of another one, male nucleus 2? Since we said pollen grain having 1 nucleus will undergo division and form 2 nuclei, male nucleus 1, male nucleus 2. Male nucleus 1 fuses with the nucleus of the egg and forms zygote. What about male nucleus 2? Now this fellow fuses with polar nuclei. This nuclei, there are 2 nuclei. So polar nuclei, or we say central cell. And this results in 3 nuclei fusing together forms pen, primary endosperm. And this formation of primary endosperm, this is triploid, whereas zygote is diploid, 2n. So formation of zygote, 2n is the fertilization 1. Formation of triploid pen is fertilization 2. So, since there are 2 fertilizations taking place inside the angiosperms, we call this as double fertilization. So, this is how fertilization 1 plus fertilization 2 results in double fertilization. So, pollen grains are dispersed from the anthers and carried by wind or other agencies to the stigma of a pistil. It is called pollination. So, pollination is a must. Pollen grains germinate on the stigma and the resulting pollen tubes grow through the tissues of stigma, style and ovary. So, there are many pollinating agents which help in transfer of pollen grains from anther to stigma of the flower. And then the reproduction, as we said, there are two. One is the zygote formation resulting in syngamy, another is the